This is Fungi by Jordan and Ryan. Fungi is eukaryotic, which means that there is a nucleus present in its cells, and it's non-vascular, which means that it doesn't have certain channels that allow it to transport water and nutrients throughout the structure. It's also non-modal, so it doesn't move, and heterotrophic, which means it gets its energy by eating other plants and animals. Fungi is unicellular, which means it only has one cell, and their cells have a cell wall, but it's not classified as a plant. It's classified as its own certain group. This is because it doesn't have chlorophyll, which allow it to do photosynthesis. Fungi also have a very small nuclei, which doesn't make it a plant cell either. Fungal cells perform biosynthesis chitin, which is the production of a polysaccharide called chitin, which acts as a protective covering for fungi. For reproduction, they reproduce by the means of spores. Spores are reproductive cells that act similarly to seeds with plant reproduction. They reproduce in 3 different ways, depending on the species. Sexually with ascospores, basidiospores, and cospores. Asexually with conidia, zoospores, and sporangiospores. And then vegetative reproduction through budding, fission, and fragmentation. The picture in the bottom right corner is of a basic mushroom life cycle where the spores are shed and they germinate into the hyphae. The hyphae meet and build the mycelium, and then the hyphal knot is formed, and lastly, the chlamydia is formed. Also, the picture in the middle is a picture of a basic fungal cell showing the unique cell wall. For growth, fungi tend to grow best in acidic environments like plant, animal surfaces, and soil. Saprophytic fungi tend to grow best in 20 to 30 degrees Celsius, and parasitic fungi tend to grow best in 30 to 37 degrees Celsius. For growth, they usually absorb nutrients from the environment around them. That's how they grow. And they store food in the form of starch called glycogen. For texture and size, it's all dependent on the species. Some fungi are macroscopic, including like mushrooms and molds. Some fungi are microscopic, like most yeasts. And depending on the species again, it's either a powdered or a fuzzy colony.
Alice is a ubiquitous fungi, which means it's commonly found. It also grows in decaying matter. The picture shown is an example of a Microalis. Entomothoralis is a parasite to insects that can kill and disable them. The picture shown is the yellow coating on the insect is acting as a parasite and it's harming them. The origin is the phylum Zygomycota is a division of kingdom fungi with 1,060 known species that are mainly land organisms. The reproductive habits, Zygomycetes reproduce by the fusion of 2 different reproductive cells called sporangiospores. Sac fungi are known sometimes as Ascomycota. The origin is phylum Ascomycota is a division of kingdom fungi with 32,000 known species. The habit, sac fungi can be found in all ecosystems all around the world. Hyphal fragments, which are small pieces of fungal structures, can be found in the atmosphere, freshwater, and saltwater. If there are hyphal fragments being found, it may indicate there's active fungal growth nearby. Reproductive habits, sac fungi produce condia and reproduce by detaching and spreading when matured. Condia is found at the tip of a hyphae. Ascospores can be found in the sac called ascos. Pigmentation, sac fungi have a melanin pigment in the hyphal walls, so the outside pigmentation tends to be brown or black. These are 3 examples of sac fungi you can find. There's a cup fungi, Brewster yeast, and truffle. Club fungi, or better known as Basiomycota. Phylum Basiomycota is a division of kingdom fungi with 16,000 known species. Reproductive health habits, club fungi tends to develop a dikaryotic condition early on in life. This is when a cell is not diploid or haploid, but has separate parent nuclei in the vegetative cell, which are a form of reproductive cells. Club fungi can reproduce by producing asexual spores called basiospores, or by fragmentation of mycelium, which is when the mycelium breaks off into pieces and grows into a new mycelium. The picture, you can see a small fragment of mycelium latching onto another, forming a new larger mycelium. The higher club fungi produce large fruiting bodies. Some have beneficial relationships with conifers and hardwoods. Some decay wood and cause tree disease. The lower club fungi has roots and smuts and no fruiting bodies. These are 3 examples of club fungi. There's mushrooms, puffballs, and rust.
So for evolutionary history, there is the interspecific hybridization, which is how the fungal DNA exchange originated. So this is when the crossing of 2 species from the same genus happens. Early fungi spread from the water to the land when it lost its flagella tail, which allowed it to propel for the water, and they dispersed their spores and reproduced in the terrestrial ecosystems. They've also been evolving for over 1 billion years. So there is a lack of fossil evidence for fungal structures because they are so hard to preserve and most of the time are microscopic and hard to find consistently. However, recently there was a massive fossil structure found depicting a prototaxite, which is now extinct. Prototaxites lived 350 to 420 million years ago and were believed to be the largest land-dwelling fungal structure on Earth. Charles Huber was a paleontologist and in 2001 discovered that the prototaxite was actually a fungus after it was believed to be a conifer because of its height for so long. In 2007, it was widely accepted that it was a fungus. The kingdom fungi is very diverse. It developed into 5 separate categories with over 100,000 species. Some of the categories are Chytridiomycetes, Zygomycetes, Glomeromycetes, Ascomycetes, and Basidiomycetes. So the fungi tend to get their nutrition in 3 different modes. So saprophytic fungi, which can be the Rhizopus, Penicillium, and Aspergillus, get their nutrition by feeding off of decayed organisms. Parasitic species like Taphrina and Buccina tend to get their energy by feeding off of living organisms, but harming and killing them in the process. Symbiotic species like lichens and mycorrhiza get their nutrients through symbiotic relationships, which are mutually beneficial for both species. Predators can be deer, birds, and other animals, squirrels, and humans, and most of the time tend to eat certain mushroom species. The common defense strategy for mushrooms is chemical defense. This is when they release a certain toxin that can impair the growth, development, and viability of the predator. So you can see in the picture in the bottom right of a fox eating a mushroom. And then in the picture in the bottom left is a ladybug with a parasitic fungi. So their roles in the ecosystem, they can cause life-threatening disease for plants, animals, and humans, but they are also the best degraders of organic matter and play a massive role in the nutrient cycle as decomposers. They have many symbiotic associations with plants, helping both species survive in the environment, but they also act as a predator, pathogen, and parasite in their ecosystems, which balances out the population. So our topic of interest was fungi and plastic degradation. So most fungal species have plastic remediation properties, meaning that they can be used to reverse environmental damage created by overflowing plastic waste. The oyster mushroom is a good example of this fungi with these properties because they allow the plastic to go in and not be stored. So they store the compounds that are necessary for them to live, which means they don't store broken down plastic, so that mushroom is still edible. And the oyster mushroom is an example of this being studied. The University of Edinburgh right now is using a lab engineered version of E. coli that can transform particular molecules from PET plastic terephylactic acid into a vanillin flavor used in food. PET is a really harmful type of plastic that's usually used in plastic food packaging and plastic bottles. The study is still in its earlier stages, but essentially the goal is to use lab engineered enzymes from fungal and bacteria matters to convert PET materials into biomaterials that can feed humans and animals and be used in antibiotics to overall reduce plastic waste. So I think that this is a really good solution for the plastic waste problem because by itself, plastic takes 400 years to break down. But with the help of fungi and bacteria, it can only take up to a few months. So the first picture is an example of the enzyme that the University of Edinburgh is working on. The second is an example of the oyster mushroom. And the third is a picture of a landfill with depicting the plastic waste issue. And that is it.